Okay, so thank you very much, Udi, and hello everybody. It's both a great pleasure and an honor to be here today. And it's an honor also, and I want to thank Udi to be on the Scientific Advisory Board of the BLAVATNIK Center for Drug Discovery. Even though, as Udi said, I spent most of my career, let's say the past 25 plus years in the pharmaceutical industry, my heart is with academic research, especially at Israeli universities. And as it happens, there is a person in this room who was on my thesis board when I was a student at the Hebrew University. So this is especially an interesting coincidence. So I will start. I will give you some thoughts and some ideas about what I call promises and challenges of academic drug discovery, a view from the industry. So I don't want to be patronizing, but I would like to share with you some of my experience with academic and industrial drug discovery and how I think the two can actually go together and be productive in a really collaborative way. Computer. So academic drug discovery presents both opportunities and challenges. Is the outline of my talk. I'm going to start with an introduction saying why I think there are both opportunities and challenges. I'm going then to talk a little bit about hurdles and misconceptions that exist about drug discovery. And I'm going to briefly describe the drug discovery process as it is practiced now in most pharmaceutical companies. And finally, I'm going to give you what I think is a recipe for successful collaboration between academia and industry, which I think is very important. So I start with this slide, which is taken from a review that appeared in Nature Reviews Drug Discovery, actually a long time ago, 2004, but the numbers have not changed a lot, unfortunately, in the last few years. So success rate for new molecules entering clinical trials is not high at all. You can see here in the different therapeutic areas, there are some that do a bit better than others. Unfortunately, the area some of us are very interested in, which is CNS research, features especially poorly with a success rate of about 7% of compounds that enter phase 1 clinical trials that were ultimately approved as drugs and marketed. And overall, I think the number is about 13%, 1-3. So it's not easy to make a drug. I'm going to touch upon several reasons why it's not easy to make a drug and how actually we can try to overcome the hurdles. So this is the drug discovery process. Traditionally, it starts from target identification and validation. It's typically done in academic labs. Hit identification, lead optimization through medicinal chemistry, preclinical development, so that's mainly toxicology and preclinical toxicology studies and formulation and process chemistry, and then clinical development, phase 1, phase 2, phase 3, and eventually we all hope there's going to be approval of the drug and the drug is going to be manufactured and marketed. Now, this area here is traditionally known as the Valley of Death, and the reason for this is that on the way from academic research to pharma, there are a lot of corpses. And these corpses, have been victims of several processes that I'm going to describe to you now. So what I think, the major so there is a quotation here that I found very interesting. Academics do not have the funds, and pharma cannot risk funding project failure. So that's basically the reason for the Valley of Death. For us, it's too early. Many projects that we see in academia are too early, and actually we now have the NNE grant process that is going to be announced, and there were many very, very interesting projects where we will be unable to fund most of them because we are looking for things that are actually sort of relatively easily translatable into a clinically valid application, and this is not always easy to find. Now, the reason why it's so what is this gap? So academia is very productive in studying disease pathways and in discovering targets, and industry is very good in developing drugs. So there is this region in the middle that is mainly target validation, lead identification, and lead optimization, where really there is a need to find tool molecules to identify and optimize leads, to provide preclinical proof of concept of some of these great ideas that are being developed here, to study pharmaceutical and drug metabolism and pharmacokinetics properties of the molecules, and study some early exploratory ways to study toxicological liabilities, and also to work on early biomarker development. And I think it is possible to close the gap. This is why we are here today. I mean, this is one of the reasons this center was established. I think there are two ways really to close the gap. One is by doing academic drug discovery, and the other is, and these actually go together in my opinion, is for academia to closely collaborate with industry. So what are the opportunities for academic drug discovery? So I don't need to convince you that there are many opportunities to do drug discovery in academia. The most important ones, I think, are to leverage the academic strengths, so intellectual novelty, you discover discovery of genes, targets, mechanisms, pathways, and also technology innovation. You mentioned, Udi, before, the many faculties that are present here and the many different disciplines that are present at Tel Aviv University. So this is really a unique situation and an enormous opportunity to combine different intellectual strengths, really, in order to come up with new ideas, with new technologies, and to combine these. And also, the other thing is to leverage the academic independence. So so you are completely free to do almost whatever you want and almost whatever interests you, not like us, to do curiosity-driven research, which is really, I think, the essence of academic research, is to be curious and to do what I call here it isn't very likely, but it would be great if science. So this is something that we cannot do, and we don't traditionally do. We don't do anything that is not very likely, or that we don't think is very likely to happen. Even if it would be great, we just cannot do it, but you can. And so you have a freedom to work on projects of low profitability also. You mentioned rare diseases. One could also add maybe neglected diseases. So you can do all of this. And there is, even though you might sometimes think that this is not the case, but believe me, if you work in industry, you think that in universities there is a lack of boundaries and bureaucracy. which is very good for research. Now, there are drugs that have been discovered in academia, not many, but there are some important drugs that have been discovered in academia, about 30 in all. I'm including only, let's say, novel type of drugs, but not all are really completely novel. Some are, for instance, 10 nucleosides or nucleotides, 10 prodrugs or analogs of known molecules. So these are, follow sort of different type of logic. Nucleosides and nucleotides are usually not the result of structure-activity relationship studies. They were discovered sort of by chance. I mean, you play around with many different modifications until you find one that somehow works. And prodrugs or analogs of known molecules, so it's more of a, it's not entirely novel. However, and many have been sort of chance findings, not based on SAR, as I just said, or not designed for final targets. However, there are also examples of really, truly innovative drugs that were discovered in academia. I list some examples here. For instance, pemetrexed, that is an antifolate that discovered in the 70s at Princeton University, that was then developed by Lilly. Vorinostat, or SAHA, compound that was that was found in an HDAC inhibitor that was discovered at Columbia and Memorial Sloan Kettering, that was in-licensed by Merck and became the first histone deacetylase inhibitor for the treatment of certain types of cancer. Zanamivir, which is a neuraminidase inhibitor that was discovered in Australia, and that became one of actually the first, I think, anti-influenza drug by GSK, and argatroban, a thrombin inhibitor, discovered at Kobe University. Now, what is also very interesting is that there are a number of CNS drugs that were discovered in academia. And the very interesting fact, and the reason I show this here, is that 4 out of 10 CNS drugs were actually discovered in Israel. So it's particularly, I think, it's a reason to be proud. And it's a very interesting fact that 4 out of 10 CNS drugs from academia were discovered in Israel. And these are, of course, Copaxone, that made really the success of TEVA, Rasagiline, which was also a very important drug for TEVA. So Copaxone obviously discovered at the Weizmann Institute, and Rasagiline at the Technion, rivastigmine, semivelin, and there are others listed there. Now I'm coming to the, let's say, not so nice part. So there are not only opportunities, there are also many challenges. There are many challenges. We said it's difficult to find drugs, and it's even more difficult to find them in academia. And some of the reasons are listed here. So drug discovery, first of all, is a very risky business. We saw the success rate before. This is a quotation I really like. One colleague once joked with me that next to show business, this is probably the job with the lowest probability that the goal of your entire career will ever happen. In show business, you want to become a superstar. And as a drug discoverer, you want to discover a useful drug. The chance of this happening is relatively low. However, it happens to some people. I'm actually one of the blessed ones to which it happened. It's a fantastic experience, because discovering a drug that actually helps to save the life of people is really something really outstanding. And really sort of provides the reason why we get up in the morning and do what we do. However, scientific and technical excellence is not sufficient. There must be a deep understanding. This is a quotation from this review in Expert Opinion on Drug Discovery, very from this year. There must be a deep understanding of the many risks and pitfalls involved. No, sorry, this is the quotation. This is my quotation. There must be a deep understanding of the many risks and pitfalls. It's very important to understand it and to find ways to mitigate against this. And adequate strategies must be put in place to well evaluate such risk and to mitigate. And it requires multidisciplinary collaboration, which Udi mentioned exists in this institution. This is very important. So the hurdles for academic drug discovery in particular are that generally, and I say generally because obviously one cannot, I mean, there are exceptions, but in general, in universities, there is limited medicinal chemistry expertise, and there is limited capacity for synthesis of large number of compounds. Usually, drug discovery programs in pharma industry, thousands of compounds are synthesized, which is not always possible in an academic setting. There is limited expertise for assay development and assay automation. There is limited drug metabolism and pharmacokinetics capabilities. There is limited bioanalytical capabilities like LCM, SMS, automated sample preparation, and there is limited integration of capabilities. And even though steps are being taken in this center to really overcome some of these hurdles, still, some will, a limitation will remain, and it is important to see how to really mitigate against this. And this is actually is from a review from last year in Nature Reviews in Drug Discovery, where they propose several ways to actually mitigate the risk of academic drug discovery, which basically is, so you have here in the middle a academic drug discovery institute like this one. And that actually should interact with external partners, industrial, obviously funding consortia, contract research organizations, and also other academic researchers, other academic collaborators, in order to mitigate some of the, to bridge the gap and to solve some of the limitations that I mentioned before. This is also from a review last year, maybe the same one. They give a sort of recipe of what academic drug discovery should do, and I think some of these points are quite important. So institutional support for translational research, a culture of collaboration. The institutional support is very important because in drug discovery, so just as an example, I mean, the people who are going to do the work here are PhD students mainly, and PhD students need to publish. Now in drug discovery, you don't always publish immediately. If you publish, you sometimes burn your chances to use your discovery. So there must be, so for a student who works in an academic drug discovery center, there must be an institutional mechanism by which this work is being recognized, even if it does not immediately yield to publications. So for a student that does drug discovery must be able to get a PhD, even if he has not published a certain number of papers in peer-reviewed journals, because the publication might come maybe 2, 3, 4 years later. You have to play to academic strengths, and this is obvious. You have to mine the institution for projects, so don't wait for people to come to you with interesting projects, but you have to go look for them, because sometimes people, scientists, don't even realize that what they are doing might be useful and might be applicable and might actually be the subject of further exploration in the drug discovery sense. Fearlessly evaluate the science, define success broadly, consult external experts, reduce hurdles to industrial collaborations. And of course, I'm personally very interested in this one, and develop a long-term stable funding strategy, but of course, you know all of this. And I'm saying it's not sufficient to find potent drugs. It's actually relatively easy nowadays to find potent drugs. Medicinal chemists have become very, very good, and there are many methods to develop extremely potent compounds, but the compounds need to be not only potent, they need to be selective, they need to be likely to work in humans, so they should have appropriate pharmacokinetics, they should show target engagement, lack of toxicity, and you need to understand at a very early stage how to prevent late-stage failure. And drugs fail for multiple reasons and in every phase of development. This is something that is not really appreciated by the public at large. There are many reasons for drugs to fail. Drugs fail all the time. One of the reasons drug development is so expensive, it now costs on average $1.2 billion to develop a drug. $1.2 billion. The reason for this is that this takes into consideration all those that failed. So if you take this into consideration, then drug development becomes extremely costly. So some of the reasons are insufficient potency, some of the reasons are lack of efficacy in animal models, inadequate PK, lack of efficacy in the clinic, also safety reasons, toxicity of all kinds, general toxicity, genotoxicity, chemical reactivity, metabolic interactions, etc. And there are some misconceptions also that I think we should fight together. There are misconceptions that we sometimes see, let's say, in academia, and that I think one of the roles really of the BLAVATNIK Center is going to be to educate the academic community about these misconceptions. And some of these misconceptions are listed here. So for instance, that efficacy in preclinical model predicts efficacy in humans, that human PK can be simply extrapolated from rodent PK, pharmacokinetics, that, for instance, total tissue levels of a drug represent pharmacologically active concentrations, that lack of acute toxicity in mice or rats means that the molecule is safe. I heard this argument so many times, but my compound, I give it to rats and they are fine. So it seems safe. Okay, so this is a fallacy it's a dangerous fallacy to make. And finally, this is one of my favorite ones, but it's become almost a sort of a joke with me, that peptides, and I come from the peptide world really. I did my thesis on peptides. I worked on peptides also later on, also at Merck, that the idea that peptides are relatively easy to develop as therapeutic agents. And so I'm going to start actually saying one word about peptides. So peptides have many advantages. They're easy to synthesize. They're usually highly potent and selective, very highly potent and selective. You can make peptides with picomolar activities against targets. There are no metabolic liabilities, metabolic in the sense of drug-drug interactions, reactive metabolites, etc. However, there are also many very serious limitations. So they have high clearance, they have low membrane permeability, and they are not orally bioavailable. And however, so I don't want to be completely negative. There are many opportunities for peptides. Generally, peptides are a promising class of drugs if oral bioavailability is not required. There are examples like that. So if you need an injectable, and there are many good examples of that, like insulin, for instance, then peptide is great. The target must be extracellular, because a peptide will not penetrate inside the cell. Even though there have been attempts to make peptide cell penetrant, these attempts have not been entirely successful up to now. And if the target is in the periphery or in regions of leaky blood-brain barrier, then you can target it. If it's in the brain, it's very difficult. The plasma half-life can be improved, so this is feasible. And peptides, I should say, are in an academic drug discovery setting excellent tool molecules. So it's great to have a potent peptide to interrogate whether your target has biologically relevance or not. If you don't think it should be a drug that is going to be marketed, but you just want to use it as a tool compound for research, peptides are almost perfect. Okay, so a few words about the importance of target validations and PK/PD studies. So the lack of efficacy, this is very interesting. Also, this is not well known by everybody, that the major reason for failure of drugs in phase 2 clinical trials is actually not safety. It's not pharmacokinetics. We become very good at predicting human pharmacokinetics. There are strategic reasons, so companies, deprioritizing some projects over others, or realizing, that there is a competitor, or that there's, the commercial potential is not exactly what they thought it was, or they are moving into other therapeutic areas, or they abandon it. The major reason is lack of efficacy. Half of drugs fail in phase 2 because they just don't work. And I'm going to here give a few examples of the poor translatability of animal models. And these examples are all from CNS, from the CNS field, but I think it's relevant because many of you probably are working in the CNS field. Just a few examples of poor animal models. So this here is Huntington's disease. So there are mice models, and all of these molecules that are listed here is in a review from Hersch and Ferrante from 2004, some time ago. They listed all these molecules that actually work in mouse models of Huntington's disease. So they are therapeutic. They cure the mice of Huntington's disease, so to speak. And Hersch and Ferrante commented in this article, because these are actually very good models at many levels of analysis, it would be surprising if some compounds benefiting the mouse models did not prove to benefit humans with HD. They said this in 2004, and they said some of these will certainly work. So some of these went into the clinic, and they are marked here by red stars, and failed. Most of them, or almost all of them. Okay, no benefit. And there were additional compounds that failed. They were not listed on this table. They are listed here. So the mouse model for Huntington's disease did not prove to be particularly predictive. Same thing for Alzheimer's disease. So many people here work on Alzheimer's disease. No need to tell you that 244 agents were tested in clinical trials between 2002 and 2012, and one was approved, 1 out of 244. So that's a success rate of 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. 0.4%. depression, all of these compounds, very nice rational and effects in animal models of depression, all failed in the clinic. Stroke, 1,026 experimental treatments in acute stroke, nothing in the clinic. And a few words about pharmacokinetics. So pharmacokinetics, I'm over time, I guess, right? So okay, I'm going to hurry. Oral bioavailability in animals does not translate to humans. This is animal bioavailability, this is human bioavailability of the same compounds. Rodents, dog, primates, nothing matches, nothing. So if your compound is orally bioavailable in animals, it does not mean anything. So if you say, I have tested the drug, this is orally bioavailable in mice, okay, that's fine. It's important if you're going to dose your compound to mice in order to see whether there is a pharmacological effect, it's important to know whether it's bioavailable or not. But it's not sufficient to make conclusions about human pharmacokinetics. Same thing about clearance. So the previous one was oral bioavailability. This one is about clearance. You see absolutely no correlation between animal clearance and human clearance for a series of compounds in rat, dog, and monkey. There is no correlation. So how are we going to get out of this? Okay, some people say, okay, you can do allometric scaling based on body weight differences. So this works very well for proteins, you see here. This is the ratio between predicted and the percentage error, actually. So 0 error for proteins, this here for small molecules. Okay, so up to, 5,000, 50-fold error in prediction using allometric methods. So how can preclinical studies that help address these issues? Because it is possible to address them. And I'm going to go quickly to. So first of all, it's possible to make mouse studies work. And this is a paper published in 2008 in Nature, I think, yes, in Nature, about the SOD1 model for ALS. And what they did is they tested, I don't remember the name of the foundation, Neta, help me, LSTDI, retested all the compounds that had been published in the scientific literature to work in the SOD mouse model. They retested them using very strict defined standard conditions and, blinded and using animals of both sexes and the right number of animals and the right statistics, etc. And they found these were the results that had been published, change in survival observed. And these were the results that were obtained when the study was done correctly. And so basically, none of these compounds worked. And actually, all of these failed in the clinic. Some of them went to the clinic on the basis of these data, and they all failed. Which makes us think, that actually the only one that sort of worked a little bit, both in the clinic, it's actually the only drug approved for ALS, and also works a little bit in the animal model, which might mean that perhaps the SOD mouse does accurately predict efficacy, contrary to common belief. And now, one word about biomarkers. Biomarkers are very important. They answer the following question. Does the compound get to the site of action? Does the compound cause its intended pharmacological effect? Does the compound have beneficial effects on disease or pathophysiology? What is the therapeutic window? And finally, they also identify responders. Now, this is something that one should absolutely avoid to do, and instead use biomarkers to study target engagement. So you should not test in vivo activity and then do mechanistic studies in pharmacokinetics, but you should first do mechanistic studies in pharmacokinetics and then test in vivo activity in order to avoid unpleasant surprises. And you actually should base your decisions based on biomarker data. So you absolutely need the answer to the question, does my drug engage its target in vivo? So does it interact with an enzyme? Does it bind to a receptor? And in vivo. And there are methods to do this. If the answer is no, that means that the drug is not potent enough or does not reach its target, so you can continue to try to optimize it. If you find that it does, then you ask whether it has an effect on a pharmacodynamic biomarker, which is also called in human study proof of biology or proof of concept light. Example would be, an effect on cholesterol, an effect on glycemia. If the answer is no, that means that your concept is not valid. So if your drug engages the target but does not produce a desired effect on a surrogate biomarker, that means your concept is not valid, and you can actually stop development. And what we call walk away with confidence. If you do not do this, but you continue, you will fail. You will certainly fail, but you could have wasted, 5 years of development and a lot of money. If the answer is yes, then you proceed to full efficacy studies. And this is some example actually from a Pfizer analysis. They analyzed 44 programs between 2005 and 2009 for whether they had the requirements for targets, exposure, target site, target engagement, and expression of pharmacology. And really, those that did not all failed in clinical studies. And those that did, all of them achieved actually proof of mechanism. I'm going to skip this because there's no time. I also want to show you that actually it is possible to predict human pharmacokinetics, not by just doing rat or mouse PK, but by doing a combination of studies. This is taken from a paper that I published in 2009. It's a prediction of human PK of HIV integrase inhibitors. I'm not going to go into the details how we did it, but it is possible to do it. We just need to invest more time and research in order to do it, not just doing simple PK studies. And this shows that the prediction actually was extremely successful. We had 4 compounds in the clinic. 3 were HIV integrase inhibitors, 1 was an HCV polymerase inhibitors, and as you see, the predicted values were very close to the observed values. So they were not like 5-fold, 10-fold differences. It is possible to predict if you just do it the right way. And this is also just briefly to show you that the discovery funnel, while once upon a time it was sequential, so you tested sequentially activity, pharmacokinetics, in vivo potency, in vitro toxicity, etc. You now all of this in parallel in order to time optimize the process and also to solve multiple issues in parallel. So it's not base yourself on activity, but base yourself on an optimization of all the parameters in parallel. And I think I just want to give you what I think is a successful recipe for collaboration with industry, because I think that the collaboration with industry will really help to progress the academic compounds further into development. I think it's important to engage at an early stage, so at the stage of target discovery, of target validation, of lead identification. Each partner should really contribute unique experience and capability, so the academia obviously the things you are good at, and industry with medicinal chemistry, assay development, DMPK, toxicology, and pre-formulation studies. And all of this in order to discover together the next generation of compounds that actually made the success of TEVA. Thank you very much.
Thank you very much, Ralph. I think it was a wonderful presentation, and this is truly the combination both of the vision as well as the practicality of drug design. And I really would like to congratulate TEVA on bringing you back to Israel as a senior vice president after so many years in experience with international innovative pharma company. I think it says something about the commitment of TEVA to new innovative drug. Due to the lack of time, we'll break to a coffee break. We also have the board meeting of the center later. I'm sure that Ralph will be happy to answer questions during the coffee break, and as I know him, he's a very approachable person. So if somebody has a private question, please contact Ralph. Thank you, and see you in 20 minutes from now.